Good morning. So I'm Geeta, Swamy. I will just introduce myself briefly, even though I was a panelist on the first session. So I am an obstetrician, gynecologist, and maternal fetal medicine specialist at Duke University, and I'm really pleased to be here with you all. I'm going to be moderating our next session entitled Stakeholder Perspectives on Challenges and Opportunities to Optimize Post-Approval Pregnancy Safety Study Types and Designs. Our speakers this morning, I'll introduce in just a moment, but in this session you're going to hear their thoughts on factors that have the potential to enhance the generation of data from drugs and biologics in pregnant individuals. You're going to hear from individuals across academia as well as industry. They're each going to have a presentation, and then we'll have a discussion amongst our panelists. So I'm just going to introduce everyone who you're probably hopefully mingling through the morning already, and it's great that we're able to be here in person together as well. So Dr. Christina Chambers is a perinatal epidemiologist and professor in the Department of Pediatrics and School of Medicine at UC San Diego. She's the PI of the mother-to-baby pregnancy studies and has led a number of national and international complex longitudinal cohort studies, as well as clinical trials focused on environmental causes of birth defects, adverse pregnancy outcomes, and childhood disabilities and conditions. Dr. Jessica Albano is a pharmacoepidemiologist by training and is the Vice President of Epidemiology and Analytics at Syneos Health. Dr. Albano serves as a PI for the Antiretroviral Pregnancy Registry, which is an international collaborative. That's the Pregnancy Exposure Registry, and that's been actually ongoing for over 30 years. Dr. Christine Olson is an OBGYN trained medical officer at the CDC's Immunization Safety Office. She's served as an expert consultant in infection control and prevention measures in maternity care settings for the World Health Organizations and on multiple federal collaborative work groups. She has been leading the CDC's COVID-19 vaccine pregnancy registry for the past few years as well. And Dr. Elyse Kharbanda. Elyse is a pediatrician by training and is the executive director for research at HealthPartners Institute, which is a health system-based research institute. And her work focuses on safety of vaccines used in pregnancy, primarily through her work with the Vaccine Safety Data Link. So I'm going to take a seat and turn over to Dr. Chambers to get us started.
Thank you. Okay. Thank you so much, and I appreciate the opportunity to be able to speak on this panel. So I'm going to, as said, I'm going to give a little bit of the perspective of pregnancy registries from the mother-to-baby point of view, and a number of points that I'll make today Dr. Sahin and others have already made. So mother-to-baby, for those of you who are not familiar with this, is a U.S. and Canada-wide network of counseling services provided by the nonprofit organization of Teratology Information Specialists, or OTIS. These were established in the 1980s, and there are about 14 services currently funded by HRSA, just recently by CDC and other state sources. And these services provide a service, individualized evidence-based information to pregnant and lactating persons and healthcare providers and the public about the safety of medications, vaccines, infections, other exposures that a person may have had already or is anticipating during pregnancy and lactation. So in the last 25 years or so, because of the dire need to have information to provide people who contact us with these questions, Mother2Baby has engaged in collaborative pregnancy studies, and we conduct these studies like a pregnancy registry, an open cohort study through a single research center at the University of California, San Diego. So mother-to-baby pregnancy registries are constructed as a cohort study design, and it's, as Dr. Sahin mentioned earlier, these are a registry design that recruits exposed and then comparison pregnant persons in the US and Canada to compare the standard outcomes, major structural birth defects overall, and then we specifically review these for a pattern of malformations, as has been suggested by others. We also look for a pattern of minor or more subtle structural anomalies among infants who receive a study-related physical examination, and then we look at the other range of outcomes, including pregnancy loss, preterm delivery, growth both at birth and postnatally, and then typically over the first year of life, serious infections, malignancies, and then at least short-term neurodevelopmental screening up to 1 year of age, and some studies have longer-term neurodevelopmental follow-up. So the schema for the overall study design is shown here. Depending on what the exposure is, we typically recruit in the middle of the first trimester, about 7 to 10 weeks. And women go through an extensive telephone interview at that point in time about all of their exposures, comorbidities, pregnancy history, medical history, and so on. And then that's updated with an additional interview in the second and third trimester. And then depending on what the outcome of the pregnancy is, as soon as the pregnancy ends, there's an outcome interview. And that's the point at which we request and abstract medical records. So that's from the OB, hospital of delivery, pediatrician, any specialty physician, any pathology reports, and through that 1 year follow-up period, if there's been a suspected malformation, additional records are requested to help confirm what that might be. And then in the 1 year follow-up period, most studies have this, for live-born infants, we offer the specialized physical exam conducted by a team of 5 pediatric geneticists, dysmorphologists who do a blinded exam of the child for these subtle minor anomalies using a checklist. And this is now done typically by telemedicine since the pandemic. And then the standard follow-up is to 1 year when we get another pediatric medical record to abstract for growth and malformations, and then the neurodevelopmental screening. And then the arrow goes out because we consent people to further follow-up if necessary. So some studies go out to 5 years. And we typically recruit for a medication a cohort that has exposure, meeting the exclusion criteria, a comparison cohort that has the same underlying disease as the exposed cohort but not exposed, and then a secondary comparison group that has neither the exposure nor the disease. Assumptions, the key assumptions that have been mentioned earlier that we're functioning under are that pregnancy registries, including ours, are typically statistically underpowered to evaluate the risk or safety for specific congenital anomalies unless the magnitude of the risk is astronomical. And even if a registry is adequately powered to rule out, say, a 2- or 3-fold increased risk of major birth defects overall, that's not what we expect of a teratogen. We expect that specific malformations or clusters of anomalies would be induced, and that's what the teratogenic effect would be. So that's basically what we're looking at. So as was mentioned earlier, an initial goal is to try to rule out this, say this is not a thalidomide, this is not an isotretinoin, this is not a mycophenolate, this is not probably a methotrexate, and then maybe this is not a valproic acid, and then beyond that, depending on the sample size and if there is a signal, what we can do. Of course, this requires careful evaluation and accurate classification of congenital anomalies in the context of knowing specifically what the gestational timing of the exposure is as it relates to biological plausibility of it being causally related to the outcome, and consistency of patterns, if any, are identified. And as has been mentioned, teratogens are often associated not just with malformations, but with a range of adverse outcomes, including increased risk for pregnancy loss, growth deficiency, and possibly neurodevelopmental deficits. So using those points or keeping those points in mind, mother-to-baby pregnancy registries focus on a range of outcomes, so they all include the range that I described. They all include a minimum of a standard 1 year follow-up in each of the cohorts. We have this addition of the study-related physical exam for minor anomalies, and specifically to assess these children for a pattern, as this pattern of minor anomalies may occur more frequently than an increased risk for a major birth defect associated with the drug. So it's a way of screening for that. And the source of data on exposures, what we call the truth, it comes directly from the mother. So it's always surprising to me that what a mom is prescribed is not always what she takes, even for medications that you would think she would adhere to very closely, they often do not. And so having this internal comparator groups we think is incredibly important. They're recruited and followed in the same manner and for the same follow-up period as the exposed group. And then as has been mentioned earlier, the importance of potential confounders, as these are observational studies. We assess a wide range of potential confounders, including co-exposures and comorbidities that are unlikely to be obtainable or obtained reliably from any other source. So the limitations, as have already been mentioned, are time and resources required to do these. This is a limitation. I think we often have relatively limited diversity in socioeconomic status and race ethnicity in the sample who enroll in these studies. From the standpoint of teratogenicity, you could argue that if a drug is teratogenic, it's going to be teratogenic irrespective of your socioeconomic status or race ethnicity. But in terms of generalizability and so on for other outcomes, it's a deficiency that we're working on rectifying. We typically have small sample sizes, and then important to keep in mind that if the medication exposure is not continuous throughout pregnancy, we have statistical power issues that are exacerbated if the exposure is intermittent. So is the exposure sample size, it really becomes the sample size that's exposed in whatever the critical window is for the outcome that you're looking at. And then last, one study is only one study. So pregnancy registries are not the be-all, end-all, and never will be. They're one source of data that can contribute to our understanding of safety. So I want to say a word about our collaboration with the American Academy of Allergy, Asthma, and Immunology, what's called the Vaccines and Medications in Pregnancy Surveillance System, as an approach to addressing this issue of needing to have multiple approaches to address the same research questions. So this was established back in 2009 with the pandemic H1N1 influenza outbreak and need for safety data on vaccines that were being generated at that time. So under this umbrella, we run the pregnancy registry cohort arm, and then there are two complementary arms, a database or claims data arm run by the Harvard group. Krista Hybrex is here today, and Sonia Hernandez-Diaz run that. And then we have had a case control arm, which has been the Boston University case control study, but more recently the CDC BD STEPS. And the point of this is twofold. One is to be able to say, rather than doing this sequentially, that we'll look at a pregnancy registry and then we'll follow it with this kind of study or that kind of study if there's a signal, to do these simultaneously so that we can get to answers and interpret the complementary data as a group more efficiently and quickly. But also a second objective is that if the pregnancy registry is failing to recruit, having the claims data information to help us understand or put into context whether the prevalence of exposure is really there to support the pregnancy registry recruitment is really incredibly helpful in determining what's feasible or not. So we have produced complementary papers, for example, with the case control and the cohort or registry arm on the example here is pandemic H1N1 vaccine in Vaccine, the journal Vaccine. So on the next couple of slides, I'm going to give you a few examples of pregnancy registries through mother to baby that were so-called quote unquote successful. And really the take-home message here is that as has been said before, many of these have taken a long time to recruit. Whether it's due to the kind of trajectory of prevalence of exposure or the participant burden of participating in a registry. So people who have to make the commitment to do this over a 2-year period of time or longer and to engage in providing us with a lot of information. Multiple factors may be involved, but these are ones that have been quote unquote successful. So we met our target sample size. We typically have quite low loss to follow-up, so not threatening the validity from that standpoint. And those that were completed some time ago are in the pregnancy or product label in the section regarding pregnancy. Ones that have just been completed in the last year, we expect will go into the label, and we met our target sample size there. And then we have 2 that are just winding up in this next year. We completed enrollment for a Pfizer COVID-19 vaccine study. It met our target sample size and are completing one on dupilumab. So these are so-called successes with modest sample sizes, but met the target enrollment. these are 3 examples of ones that we might call failures. So the target sample size, despite many years of recruitment and promotion of the study and so on, was not met. We continued to recruit in the comparator groups, but as you can see here, for tocilizumab, tofacitinib, and mepolizumab, we only achieved between 11 and 34 exposed pregnancies ballpark. The last one, mepolizumab, Dr. Wurst was talking about earlier. Here's a drug that was part of this VAMPS collaboration, and we were really, this was an example of where we didn't know, as was expected when this drug was first marketed. We thought, well, maybe it'll be like the first biologic in the asthma space, that we would be able to achieve a sample size as was outlined here, and it turned out that we did not. But the claims data in VAMPS was really useful in helping us understand that the prevalence of use was quite low, and that consistent with us not recruiting as many as we had anticipated. So when a pregnancy registry works, such as ours, what are the pluses? If the prevalence of use in pregnancy is sufficient to support the pregnancy registry, we think having this ongoing open cohort platform, and I'll call it a platform because it doesn't require you setting up independently one-off a new registry from beginning to end. It's layered onto an existing platform, so it requires very little setup time. It's the amount of time that it takes to get an addendum for the sub-study approved by the IRB. Engagement with the pregnant woman leads to, we think, a high retention rate, why we have limited loss to follow-up. We have the ability to, we think, do a good job of classifying outcomes using these multiple sources of data, including maternal report, medical records abstraction over a year's period of time, and then this study-related expert assessments. We have the ability to confirm actual exposure, dose, and gestational timing by capturing this from the mother. We have the ability to acquire data on covariates that are not typically available through other data sources, including EHR, such as substance use, over-the-counter medications, fever, herbal products, as well as we have the ability to capture measures of disease severity and have these disease-matched comparators. And we have the ability to do broad and extended follow-up if needed, including responding to a signal. So I'll give you one example of a signal in the etanercept registry. We did identify a pattern of 3 or more minor anomalies that were the same ones identified in 6 children who had received the study-related physical examination. And so we followed up on that signal by having those children and their parents re-examined by a different examiner to confirm that the minor anomalies that are sometimes very subtle were actually there, and to see if they were in the parents as well. And then we offered neurodevelopmental testing face-to-face for those children, and 5 of the 6 completed it. And the result was that the parents, most of the parents also had the minor anomalies. The minor anomalies that were seen in the children were validated by the second exam, and all of the children who were tested performed within the normal range. So evaluation of the signal, we felt, led to no further concerns. If a pregnancy registry such as ours doesn't work, what then? So why did you start it in the first place? And I know that's kind of what the framework is about here. If the prevalence of use in pregnancy cannot be reliably predicted to be rare, so you just don't know, we think it's important to plan for a feasibility period. So to say, rather than saying we'll do this indefinitely and hope that we reach a sample size that's interpretable, to say after a given period of time and using other data sources such as claims data, is this really feasible as a source of data, and not to further invest in going in that direction. As mentioned, it's important to have an additional source of population data such as the claims data or EHR to confirm or refute evidence for low use. And as was mentioned earlier, and I'm glad that Kathy Wisner brought this up, we do need an approach and a systematic approach to interpretation of small numbers. So it's one thing to say you have 3 exposed pregnancies and 2 were preterm, and then what do you do with that? Nothing, there's not much you can do. But if you have 30 exposed pregnancies and you don't have 25% who have a pattern of malformation like mycophenolate or like isotretinoin, can you say this is not a thalidomide? Right, maybe you can. So coming up with some approach to being able to deal with those smaller numbers, as long as the data is of good quality, I think would help the field in general. So final comments. I think we can build on efficiencies associated with disease-based registries, and I know Jessica Albano is going to talk about this, and theirs is the perfect example of this. I'd go a big leap further and say, given the opportunity to be at the pulpit here, that we would greatly benefit as a nation from the efficiency and productivity of establishing a single US-based pregnancy registry, not just a disease-based registry, but a pregnancy-based registry to serve as a signal detection system for all new drugs, irrespective of the prevalence of use. So not having to bank on there will be enough exposures, but creating a pipeline whereby healthcare providers and pregnant women could all be part of this. And to the comment from the FDA earlier, I don't see why people who are planning pregnancy couldn't be part of that to initiate enrollment in a pregnancy registry when they're planning pregnancy with some minimal baseline data so that we actually can follow them from the time of conception throughout pregnancy. So there would be a lot of fiscal efficiencies and a lot of scientific efficiencies, I think, as well. And then in the meantime, I think it's important, and this is a broken record, that when pregnancy registries are initiated, that they do not function in a vacuum, that we need coordinated efforts across data sources. So I will end there. Thank you.
All right, good morning. It's a pleasure to be here and to talk with you today about my insights on pregnancy exposure registries from a multi-product and disease-based perspective. All right, by way of disclosures, I'm an employee of Syneos Health that runs the Antiretroviral Pregnancy Registry, which is a collaborative study jointly funded by the following list of manufacturers. So multi-product or disease-based registries can be defined in two ways, based on the patient's exposure to the specific drug or products being monitored. Usually this is without the regard to indication for treatment and includes all marketed brand and generic versions, including regardless of how they are prescribed. And then secondly, based on a patient's diagnosis of a particular disorder, including those who are either treated or untreated. In general, multi-product or disease-based registries can be a good consideration when the products are manufactured in combination, and therefore individual drug exposures can't be separated out. There could be complex multi-drug treatment regimens, again, making it difficult, if not impossible, to separate out exposures. There's a high likelihood of polytherapy, so multiple different disease-specific treatments that are given, or polypharmacy, when multiple different diseases are being treated at the same time in the same individual. The treatment landscape can be rapidly changing, for example, due to frequent new product approvals in the market. Internal comparisons are desirable, or when confounding disease or population characteristics exist. Ultimately, the focus needs to be on evidence-based data collection that better meets the needs of both the physicians as well as the patients who are making the treatment decisions. We have work to do towards expediting the accrual of patient safety data in order to inform patients and healthcare providers in a timely manner. To put this into perspective, we've heard a lot about sample size so far this morning, but if one is looking to rule out a twofold increase in the overall risk of birth defects based on a general background defect prevalence of 3%, you would need 200 live birth outcomes in order to establish a relevance of a signal or not. So that does seem reasonable, but if you put that in perspective of a more rare congenital malformation such as neural tube defects that has a background prevalence of 0.1%, you would need 2,000 exposures in order to evaluate that potential signal. So we can agree that there are very few examples of pregnancy registries that have met that threshold. And while some disease pregnancy registries may have as many as a half dozen or more distinct registries that have been launched to address these questions, and even some have ended potentially even meeting their sample size goals, it is unlikely that they were individually robust enough to provide sufficient evidence of risk. So for the remainder of this talk, I'm going to focus on the pregnancy exposure registry, the antiretroviral pregnancy registry, of which I serve as the primary investigator, and some lessons learned that can be applied to this situation. I'll start by, there we go. Setting the stage here that the APR is a collaborative endeavor with multiple stakeholders that work together for the common goal. The APR is a voluntary international prospective exposure registration cohort study. The purpose of the registry is to assist clinicians and patients in weighing potential risks and benefits of HIV treatment during pregnancy. The objectives of the registry are to provide early warning signals of major teratogenicity, to estimate the prevalence of major birth defects and compare that to the general population, and to supplement preclinical and clinical and epidemiological study data. The predecessor to the APR was the Zolidine and Pregnancy Registry, which was established in 1989 by Burroughs Wellcome. With the entry of additional antiretroviral products into the market, the registry expanded to become the Antiretroviral Pregnancy Registry in 1993. The APR fulfills post-marketing commitments for the FDA. Currently, there are 24 sponsoring manufacturers. The registry monitors prenatal exposures to 164 drugs, including 61 brand name single entity drugs or fixed dose combinations, and 136 generic versions, covering HIV treatment and prevention, including pre- and post-exposure prophylaxis, as well as hepatitis B treatments. The primary outcome is prevalence of major birth defects. Infants are not followed after birth. The registry was not designated or designed to formally evaluate premature birth, low birth weight, small for gestational age, or developmental delays. The analysis is multi-tiered, so the prevalence is calculated overall and by trimester of earliest exposure for all drugs being monitored at the drug class level, at the individual drug level, and for common drug combinations or treatment regimens. Comparison groups include the external background reference groups of the Metropolitan Atlanta Congenital Defects Program, or MACDP, and the Texas Birth Defects Registry, or TBDR. And then there are also internal comparison groups. The APR also follows the rule of 3, which specifies that the occurrence of 3 specific birth defects for any exposure or exposure combination triggers an immediate review. This is particularly useful for drugs with fewer than 200 exposures for which prevalence cannot be calculated. However, understanding weak signals that may vary over time needs to be carefully monitored and is methodologically challenging. The dedication of a potential signal requires detailed analysis to evaluate and identify this risk. In addition to comparing to the prevalence, the 2 external comparison groups, having multiple available internal comparison groups, is a distinct advantage of multi-product registries. It affords further comparisons, such as exposure to drug A in the first trimester, to exposure to drug A in later trimesters, exposure to the drug of interest in the first trimester versus no exposure to that drug at all, exposure to that drug in the first trimester versus non-drug exposure in the first trimester. Comparison 1 mimics the approach used in the APR for its primary analysis, and the other comparisons can serve as confirmatory internal comparison groups when evaluating a signal. As previously described, the APR began as a single drug registry in 1989 and has expanded over the past more than 30 years to become a multi-product, multi-sponsor registry. As of January 2023, there are nearly 26,000 women defined as prospective cases have been enrolled from 75 countries. More than 70% of enrollments come from the US and its territories, and the loss to follow-up rate is currently 11%. Given the diversity of the APR, population representativeness is a concern, and reliance on a single external comparator for risk assessments is considered risky. Based on the recommendation of the APR's advisory committee, they evaluated external comparators and added, in the absence of a US national data source, a second external comparison group, the Texas Birth Defects Registry, to supplement the original MACDP. The APR continues to evaluate other international resources to further diversify comparator populations. The APR, as mentioned, requires a threshold of 200 exposures in the first trimester before conducting a drug-specific analysis to calculate prevalence. For the newest class of ARV drugs, integrase inhibitors, which includes bictegravir, cabotegravir, dolutegravir, elbaptegravir, and raltegravir, those that have reached the threshold have taken between 4 and 9 years to accrue 200 first trimester exposures. As depicted in this forest plot, there are a total of 21,636 live births, including 631 outcomes with at least 1 birth defect, 348 of those which occurred in the first trimester. The overall APR birth defect prevalence is 2.9%, 95% confidence interval of 2.7 to 3.2%. This proportion is not significantly higher than those reported in the registry's 2 external comparator groups. There's also no difference in the internal comparisons of first trimester exposed pregnancies versus the second or third trimester. To date, there's been no concern among the individual drugs analyzed, with the exception of nelfinavir and didanosine, which have modest but statistically significantly increased risk compared to the MACDP. However, for these 2 drugs, we've not seen discernible patterns among the defects reported, and due to concerns with the safety of these drugs, they are rarely used in pregnant populations of childbearing potential. Therefore, we're not seeing new exposures, and this risk is likely not to be further refined. You'll recall I started by describing the APR as a collaborative registry, one that began by choice and evolved into a post-marketing commitment over time. Essential to the success of any complex registry is a well-defined governance structure. And by a collaborative registry, I'm referring one in which multiple stakeholders work together to meet one or more specific objectives, whether this is by choice or by mandate. The figure depicts the governance structure of the APR. It's overseen by a steering committee. It's comprised of the following groups: the Scientific Advisory Committee, which is made up of experts in the appropriate fields, including academia, government, and private practice. The advisors provide scientific oversight and review and interpret the registry data. The consultant birth defect evaluator is a member of the advisory committee. The sponsor representatives are from the manufacturing companies, and they oversee the registry management, budget approvals, and regulatory reporting. The Registrating and Coordinating Center is responsible for managing the daily operations of the registry, including the enrollment, the data collection, statistical analysis, report writing, interactions with the IRB, and the facilitation and interaction of both the advisors and the sponsor groups. The primary benefit of such a governance structure is clear separation of scientific, financial, and operational objectives. The APR Steering Committee convenes twice annually to review and interpret the analysis results. The Advisory Committee meets separately to form an independent consensus statement. In an effort to help ensure that the study results are communicated consistently and accurately, the registry requires all publications of data to include a published consensus statement as seen on this slide. The APR's ability to maintain enrollment and remain relevant over these many decades is due in large part to its internal and robust dissemination of findings. An interim report is republished every 6 months, as Layla mentioned earlier, and it's posted to the study website and mailed to participating healthcare reporters. A webinar focused on treating physicians is given annually. The results are also used in the U.S. perinatal treatment guidelines, and drug-specific data is included in the product labels in adherence with PLLRA. Now that we've seen an example of a successful collaborative registry, I want to unpack some of these lessons learned, starting with the challenges. First is complexity. Whether there's 3 or 30 products in a multi-product registry, it can be operationally and analytically complex and require a high degree of expertise to carefully plan and implement. The second is collaboration. They require agreement from companies who are competing in the same therapeutic area to work together, adopt common processes, adhere to agreed policies and timelines. In some cases, there can be fairly significant differences in the profile of the sponsor companies involved with regard to their cultures, the SOPs, their regulatory interpretations, the level of risk they're willing to assume, and resources. The challenge is to bring all the stakeholders together collaboratively to avoid delays and conflict that could put the entire program at risk. Regarding communication, all parties must respect the established lines of communication, which should be appropriately documented. Competition. Multi-sponsor registries don't happen at once, but over time they grow in size, and with more entrants coming into the market. Innovator companies are usually the first to the table, frequently responsible for the design and setup and implementation of the study. Latecomers will often have to adopt the established processes and governance, which can be challenging and make documentation a key process and oversight. There is confidentiality to consider. It's necessary to have sensitivity to the proprietary aspects of drug discovery, marketing, and lifecycle management for each of the individual sponsors, as well as direct communications that might happen outside of the registry between regulatory agencies and pharmaceutical companies. However, they may have relevance to the establishment and the conduct of the registry itself. So it's an ongoing challenge to ensure adequate confidentiality is paramount in all agreed processes. And then finally, commitment. Various stakeholders, as we've even heard across industry, may not have had the same level of commitment to any given project. It's just how business works, and there might be numerous reasons for this. Equal input and engagement should not be expected, so it's important to build an expectation and commitment requirements to join a multi-sponsor registry at the outset. So despite their complexity, multi-product or disease-based registries have distinct advantages over single-product registries. We've heard some of these already today, but they're logical in that they avoid duplicate efforts in the establishment of the registry. They form reporting lines from a perspective for healthcare providers, pharmaceutical companies, and regulators that are less complex. They're economical because resources can be pooled, and as well as budgets for multiple stakeholders. Not only are multi-product registries more efficient with their use of limited budgets and resources, but with their utilization of experts in the roles of advisory board members, as well as birth defect evaluators who can provide input for the overall program on a global scale. Multi-project registries reduce competition for eligible patients and streamline healthcare provider participation. They also offer a more robust consolidated awareness effort. Multi-project registries are methodologically advantageous through the standardization of data collection, case evaluation, and statistical analyses, not to mention the enhanced validity and power. Most importantly, to the advantages, however, it comes from the clinical point of view. Multi-product registries serve as a centralized resource for patients and physicians. They minimize the reporter burden of healthcare providers. They increase the incentive to participate by providing a single comprehensive reporting mechanism. They offer a coherent assessment of the available data, and they provide a consistent message of the current understanding regarding risk for both patients and physicians. Okay, thank you very much.
All right, I'm checking my watch, though. I'm going to switch to good afternoon. Good afternoon. Thank you for having me here to present on CDC's COVID-19 Vaccine Pregnancy Registry. I'll note, although the slide is at the end, that I have no disclosures, and the opinions expressed here are those of the speaker, not of the agency. During this session, I'll be describing the concept and implementation of CDC's COVID-19 Vaccine Pregnancy Registry, the operations of the registry, including the enrollment and data collection process, the two-phase approach used during the pandemic, and the cohort of participants, and then addressing the successes and challenges of this approach. This graphic depicts the different vaccine safety monitoring systems employed by CDC's Immunization Safety Office, which include the Vaccine Adverse Event Reporting System, or VAERS, which many are probably familiar with, the Clinical Immunization Safety Assessment Project, or CISA, the Vaccine Safety Data Link, or VSD, about which you'll hear more from Dr. Kharbanda shortly, and the two newest systems that were initiated during the COVID-19 pandemic in 2020, VSafe and the COVID-19 Vaccine Pregnancy Registry, that are currently housed together under a new team, DETECT, the Data Exploration and Technology Team. So first I'll start with a bit of history from way back in the early point of the pandemic when vaccines were first becoming available in 2020, December of that year. At that time, you'll remember that there was limited data available about COVID-19 vaccine in pregnant people, and that they had been excluded from the phase 3 clinical trials, leaving only developmental and reproductive toxicity, or DART, animal data available as the only source of information at that time. At the beginning of the pandemic, we didn't know if SARS-CoV-2 in pregnancy was associated with increased risks in pregnancy. Though it is well established that there are changes in adaptive immunity and physiology that occur in pregnancy that could increase the risk of severe respiratory viral illness. And it was reasonable to assume pregnant people may be at increased risk for severe disease and adverse pregnancy outcomes. We've seen this with other respiratory viruses, notably with H1N1. This was also likely to lower the uptake of a new vaccine product in the pregnant population, and both of these assumptions were borne out over time as the pandemic progressed. The COVID-19 Vaccine Pregnancy Registry was therefore conceived, developed, and implemented relatively quickly to coincide with the early availability of COVID-19 vaccines to help fill this information gap. It was a collaborative effort between multiple areas of CDC, including the Immunization Safety Office, the Division of Birth Defects and Infant Disorders, and the Division of Reproductive Health. The new V-Safe After Vaccination Health Checker, which was a web-based voluntary safety surveillance system for adverse events, was launched in December 2020 to capture information about vaccinees' responses to receipt of COVID-19 vaccine. We were able to use V-Safe enrollments to identify individuals who reported they were pregnant or became pregnant shortly after one of their primary series vaccine doses. And this allowed a readily available convenient sample of recent vaccinees potentially eligible for the pregnancy registry to follow in fairly real time, with enrollment in the registry beginning in January 2021 and continuing to enroll those who had enrolled in V-Safe by June of 2021. The initial goal of this pregnancy registry was to monitor for adverse outcomes of interest in pregnant people receiving COVID-19 vaccines in a systematic and fairly rapid way to identify early safety signals, characterize the safety profiles of COVID-19 vaccines in pregnant people, and supplement the existing passive and active surveillance systems. The initial planned framework included up to 5 phone interviews during and after pregnancy, collecting participant-reported data, medical record acquisition and abstraction for those who consented and who reported outcomes of interest, analyzing the self-reported data initially by chronologically available outcomes. This required a clinical review step for some outcomes, such as stillbirth and birth defects, and a plan for more robust analyses later after medical record information was available for confirmation of and more detail about outcomes of interest. This slide lists the planned outcomes of interest that the registry would be able to capture by obstetric and neonatal and infant categories. Those in bold are outcomes for which we determined medical records would be most helpful and were therefore requested. I'll next describe the enrolled cohort resulting from this process and the data flow and operations. So this graphic depicts the process of enrollment that again began in January 2021 and included those who reported a pregnancy into V-Safe between December of 2020 and June of 2021. Eligibility screening ensured that participants were 18 years of age or older, spoke English or Spanish, and were pregnant at the time of vaccination or became pregnant based on their LMP within 30 days of receipt of vaccine. During phase 1, the interviews included questions about demographics, health during the pregnancy outcome, information from the birth hospitalization, and postpartum and infant health through 3 months of age. Participants were asked to consent for medical record review. In phase 2, the last rectangle on this slide, conducted 15 months after the end of pregnancy, participants who were not lost to follow-up and who had not opted out of future follow-up included questions about both participant and infant health, with the primary focus on infant health, particularly on birth defects ascertained during the first year of life, consistent with how routine birth defect surveillance is conducted. On this slide, you'll see that over 123,000 V-Safe participants, ages 18 to 54 years, were identified as potentially eligible for the pregnancy registry. In May 2021, a pregnancy follow-up survey was sent through the V-Safe platform to confirm pregnancy and to better identify likely participants among potential participants who had not yet been reached. Those who had responded that they were or had recently been pregnant and gave permission for contact were called, while those who indicated that they were not pregnant around the time of vaccination or were not interested in participating in the registry were removed from the list of potential participants. Overall, about half of participants who reported a pregnancy into V-Safe from December through June were called. This is shown in blue in the pie chart on the left. About 32% were unreachable, which was our biggest barrier to enrollment, making contact. Among the 20% we were able to reach and assess for eligibility, 94% were eligible and enrolled, bar on the right. A very small proportion of those eligible actually declined. This graphic shows the characteristics of over 23,000 total eligible and enrolled participants who reported at least one pregnancy into V-Safe between December and June. Most received one of the mRNA vaccines. The majority self-identified as non-Hispanic white. The mean age at first vaccination was 33.5 years old, and 45% identified as healthcare personnel of some type, which was not surprising given the stage rollout and prioritization of vaccination and vaccine distribution early in its availability. This graphic demonstrates the timing of the earliest vaccination during the periconceptional period or during pregnancy among our participants, with distribution seen across the spectrum, and over half receiving their vaccines in the second or third trimesters. You can see in this graphic that those in the registry who reported receiving the monovalent booster dose of vaccine during pregnancy, depicted in blue, were, as expected, those who had received their primary vaccine or primary registry eligible dose of primary series vaccine in the early part of pregnancy, either preconceptionally or during the first trimester. For our phase 2, or extended follow-up interviews, conducted 15 months after the end of the pregnancy, of over 21,000 phase 1 participants that were eligible, we were able to reach 44%. Similar to phase 1, the primary barrier to data collection was having someone answer the call from the interviewer. When the participant was reached live by phone, the vast majority completed the interview, nearly 97%, which was also similar to phase 1. Methods used to optimize reaching participants by phone included having an assigned caller ID to the interviewer caller line, following call attempts with text with callback information, including asking for convenient times to try to reach the participant again. The very overly simplified planned data flow for phase 1, which is similar for phase 2, is depicted here, with the interview data on the left generating preliminary early outcomes, such as for reported spontaneous abortion or miscarriages, followed by acquisition, abstraction, and review of medical records to enhance the interview data, with this more comprehensive data set of interview and medical record data then producing more detailed final outcomes. On the right, you can see the insertion of the clinical review work involved in reviewing and better understanding the interview data and its corresponding medical record data. This was of particular importance for birth defect adjudication and categorization. One note, particularly about one of the most important outcomes of this work, birth defects. The process for this has been fairly labor-intense and involved. We classified all reported fetal and infant health conditions as major, minor, possible, probable birth defects, or as not a birth defect. All birth defects were then coded using the MACDP process. This is what CDC uses for its routine birth defect surveillance. All pregnancies with any outcome, including live birth, stillbirth, abortions, or miscarriages, were included. And we have now, through phase 2 interviews that have just been completed, included any birth defects identified and reported through 15 months of age. I'll move now to the registry's accomplishments and challenges. In the interest of time, I'll not be presenting specific results, as that is not the focus of this meeting. Links to the publicly available data from the registry are provided at the end of this presentation in the resources slide. The registry allowed for monitoring of reported outcomes as the enrolled participants progressed through their pregnancies and interview data accrued. Excuse me. Proportions of reported outcomes were compared to expected background rates and monitored for any deviations from expectations. We were able to provide regular data updates to the Advisory Committee on Immunization Practices COVID-19 Vaccine Safety Technical Workgroup, or VAST. We provided public presentations at ACIP meetings in the fall of 2021 and 2022. And there were publications on the preliminary findings of the registry in the New England Journal in 2021, with the information noted about no noted increase in the rate of spontaneous abortion or miscarriage used to be able to strengthen the COVID-19 vaccination recommendations made by CDC to vaccinate pregnant persons during any trimester in August of 2021. Taking a broad view of this type of registry in the unusual setting of a global pandemic and global vaccination program, some of the advantages were its flexibility, its potential for medical record confirmation of consenting participants' self-reported data, and the relative speed for some of the earliest outcomes in pregnancy, such as spontaneous abortion or miscarriage. However, there were some significant limitations. Obviously, the cohort was biased toward the earliest eligible recipients and adopters of vaccine, which were largely healthcare personnel. It proved to be a very time and resource-intense effort involving large volumes of data from 2 sources, both interviews and medical records, and importantly was based on a convenient sample with no control group externally, which then necessitates comparison to available background rates of studied outcomes. Our real-world experience highlighted some important issues to consider in future similar efforts. We did not know what stage of pregnancy someone was in at the time of vaccination from V-safe data alone, as that data were not collected. So we were unable to order calls based on gestational age, which would have been very helpful. That meant many participants had already delivered by the time of the first interview. Over half of the participant reports of infant birth defects required medical record confirmation, not surprising, for further clarification, as these standardized questions were not effective in eliciting the type of more detailed information during an interview that would allow birth defect subject matter experts to appropriately categorize the birth defects from interview data alone. Medical record acquisition and abstraction was very resource intense and of variable quality based on the records received. And there's an important balance to be struck between the sensitivity and specificity of the information. For example, in a safety surveillance system, one would want to include possible probable birth defects, but this is not routinely done in national birth defect surveillance, making background rate comparisons with those data particularly problematic. And some outcomes are just not as easily captured as others, either by interview or medical records. More specifically, challenges that were unique to the interview data included the impact of more open-ended questions to improve the feasibility of interview completion meant that we captured a broad range of diagnoses, which was helpful for hypothesis generation, but resulted in decreased standardization. Use of text fields to allow for some participant responses resulted in coding challenges and some unusable data. Participant report is not the standard approach for complex medical conditions, which then required a robust protocol for classification to use the data for analysis. There are sometimes nuanced details needed for clinical adjudication and classification of outcomes that are simply not available from use of a standardized interview format. There was some inconsistency in how participants responded to interview questions, particularly for some outcomes such as birth defects and hypertension. For example, reporting birth defects only when asked about the infant's clinical referrals rather than as a direct response to a question about birth defects or infant medical conditions. Or reporting gestational hypertension or diabetes from a prior pregnancy as a chronic medical condition, which was information collected for confounding analyses. Another issue was that there was uncertainty if certain conditions that had been noted during pregnancy had resolved in utero or were still present after birth, which particularly impacted birth defect classification. And again, this proved to be time and training intense, which impacted the speed of available results. While medical records are often thought of as the gold standard for clinical data, there were several notable challenges in their use. Records were not always obtainable, that is, there may not have been consent provided, the records were not found, or they were not sent by the facility. There are also variable facility requirements we encountered for medical record release that delayed or prevented acquisition. Incomplete records were often obtained. For these outcomes, prenatal delivery, and outpatient infant records were needed and constituted what we would consider a full set of records. However, often not all of these records were available. There are discrepancies within the records, including dates of diagnoses, diagnostic terms used by providers, and repetitive procedures, such as ultrasounds that had different results. And there were handwritten records and lack of standardization across multiple electronic health record platforms, making use of what would seem a simple record, what I was used to, the prenatal flow sheet, sometimes more difficult than anticipated. It was often written in narrative form rather than graphic form and a bit on the confusing side to interpret. I think mostly a result of EHR use. Again, this proved to be a resource-intense area, particularly with training, re-abstraction of records, data quality checks, comparisons of abstractions, and feedback to improve the quality. I'd like to close with some thoughts about how we might approach this differently in the future for consideration. Clearly defining outcomes of interest that are ideally narrow in scope would have been helpful, particularly for interview questions. Importantly, this requires prior determination of a more limited set of study outcomes that may not be appropriate for all purposes, like a global vaccine rollout. Minimizing the number of text fields, planning specific analyses prior to implementation to guide data collection, including specific definitions of key variables such as pre-existing medical conditions that might be confounders for outcomes, recognizing, acknowledging, and planning for likely discrepancies within data sources, such as hypertension documentation in the medical record, that will require additional decision-making. The two most challenging outcomes we found were hypertension, both pre-existing and gestational for participants, and birth defects for infants. Birth defects is a complex outcome of high importance in these types of studies and often requires comprehensive medical records and interpretation for confirmation. Inclusion and exclusion for multifetal pregnancies is important. For example, how to handle fetal reduction scenarios like vanishing twins. Having clear and standardized definitions of birth defects is critical. Use of participant report alone, while fast, is highly challenging to standardize and interpret. And the use of medical records available after the first year of life is standard process for routine birth defect surveillance. So challenges in acquiring records delays data collection and results and must be factored into timelines. And finally, having an available control group is important, as reliance on background rates, especially when those may be fluctuating, such as during a pandemic, is limiting. We did take a look at some internal comparison groups based on the timing of vaccine receipt, but for the most part have been using background rates. I'd like to thank the registry participants who generously gave of their time and information, as well as the large group of people behind this effort. And this is the resources slide that has links to our publications and presentations. Thank you.
As Dr. Kharbanda is getting ready, I just want to remind folks they can put questions into Slido if you have them as they're coming up, and for our discussion for after.
Oh. Okay, thanks for having me here today to present my stakeholder perspective evaluating the safety of vaccines administered during pregnancy and the vaccine safety data link. And I just want to acknowledge that I am the last formal presenter before lunch break, so thanks for bearing with me. As a disclaimer, my work is supported through contracts with the CDC, but what I present these are my views alone and not official positions of the CDC. And any mention of a product or company name is for identification purposes only. So as an overview to this presentation, first I'm just going to talk a little bit about where I work at HealthPartners Institute. Then I'll provide background on the vaccine safety data link, including an introduction to our data structure, and provide some information on some studies that we've conducted in the past on vaccine safety and pregnancy. Then I'll provide my perspective on post-approval monitoring of vaccine safety and pregnancy. So first about HealthPartners, we are the largest consumer-governed nonprofit healthcare organization in the nation. We were founded in 1957 and we're based in Bloomington, Minnesota. As an integrated health system, we include a health insurance plan with about 1.8 million medical and dental health plan members. We're also a care delivery system based in Minnesota and western Wisconsin. We have each year about 1.2 million medical and dental patients, and since we're talking about pregnancy, I'll say we have about 12,000 prenatal care visits in our system each year with a live birth in our health system. And now about HealthPartners Institute. So we're a nonprofit institute within the larger HealthPartners organization dedicated to high quality public domain health research. The image on the left is a photo of our, or screenshot of our webpage. There are a number of investigators who work at HealthPartners Institute who conduct work related to post-approval drug or vaccine safety in pregnancy, and some of those projects are listed on the slide. But the rest of the presentation, I'm going to focus specifically on our CDC-funded work in the vaccine safety data link. So a little bit about the VSD. The VSD is a collaborative project between CDC's Immunization Safety Office, and we're now up to 13 integrated healthcare organizations and networks across the U.S., and these sites are shown on the slide, on the map on the slide. The VSD monitors the safety of vaccines in use in the United States, primarily through observational multi-site studies of rare and serious events following vaccination. The CDC was founded in 1990, and HealthPartners joined as a VSD site in 2000. Now a little bit about the data structure for the VSD. As the name implies, a critical component of the vaccine safety data link is our data structure. And for any group working in post-approval safety monitoring, working with high-quality data is key. Of the 13 current VSD sites, 11 are now data contributing sites, and as such, they create standardized data files that can be used in multi-site studies. We use a distributed data model in the VSD, so all data files reside at the site, and programs are run off these data sets with limited data sets transferred to a lead site for specific studies. This slide also shows the many files that each VSD site creates. Those in bold, the dynamic pregnancy episode file and the mom-baby linkage, have been critical for our work in maternal vaccine safety surveillance. It was around 20 years after the founding of the VSD in 2010 when we expanded to evaluate vaccine safety in pregnancy. There were many reasons for this shift, and these have already been discussed a bit. So pregnant women were generally not enrolled in clinical trials of vaccines, and when vaccine trials are conducted in pregnant populations, there's often insufficient power to assess rare post-vaccination safety outcomes. And as such, vaccines might be recommended for use during pregnancy with limited data on their use in this specific population. So it's clear that post-approval observational studies are needed, but these can be challenging. As the VSD includes data on over 3% of the US population, and we have comprehensive data on vaccine exposure, standardized data files, and access to medical records as needed, the VSD is an ideal network for conducting post-approval vaccine safety monitoring. An increasing number of vaccines are now recommended in pregnancy, and these vaccines are usually recommended for all pregnant women. So this slide provides a brief history of vaccine recommendations for vaccines in US populations. Today, the influenza, Tdap, and COVID-19 vaccines are all recommended in pregnancy, and the RSV vaccine may soon be added to this list. It's clear that the past 20 years have been marked by a substantial expansion in the number of vaccines indicated or recommended in pregnancy, increasing the importance of our work in maternal vaccine safety monitoring. So now I'm going to shift to talk about a few of the studies that we've worked on in the VSD that my group has led. So our first area of focus was on influenza vaccines. So the study on this slide, published in 2014, included nearly 60,000 vaccinated pregnancies from the 2004-05 through the 2008-09 influenza seasons. And these women exposed to influenza vaccine during pregnancy were matched to women unvaccinated in pregnancy. When using a propensity matching and analyses that accounted for vaccination and pregnancy as a time-dependent exposure, we found that receipt of vaccination and pregnancy was not associated with increased or decreased risks of preterm or small for gestational age birth. In the second study, published in 2017, we evaluated 50,000 live-born infants from 2004 to 2013 with maternal first trimester influenza vaccine exposures, and found as compared to unvaccinated and first trimester, adjusted prevalence ratio for having a major structural birth defect was 1.02, with a 95% confidence interval of 0.94 to 1.10. So a few key points related to our early work on maternal influenza vaccine safety. Overall, these were well-powered studies. We were able to evaluate a range of outcomes, and we had no safety signals. However, these studies took place before we had access to the dynamic pregnancy algorithm. We required access to birth records, which resulted in a substantial lag between when the vaccine exposures occurred and when we were able to report safety outcomes. Also, we combined data from multiple influenza seasons and ignored potential differences by manufacturer. And finally, at the time of these studies, our data sources for vaccines were limited to electronic health record and claims, and thus these studies might have missed vaccines administered at flu fairs or other community-based venues. Following recommendations for use of Tdap in pregnancy, we expanded our portfolio to explore Tdap safety in pregnancy. This slide shows one of our first studies on this topic, published in 2014. The study included pregnancies ending in a live birth from 2010 to 2012 at the 2 California VSD sites. Overall, our data was reassuring for SGA and preterm birth, but we did have a small but statistically significant signal related to chorioamnionitis, with an adjusted relative risk of 1.19 and a 95% confidence interval of 1.13 to 1.26. This raised a lot of questions, including what was the clinical significance of this finding, and even why were we studying this outcome? Was it biologically plausible that a vaccine in pregnancy could increase risk for chorioamnionitis? Our findings did not result in a change in vaccine recommendations in the United States, but we did conduct a follow-up study. Our follow-up study evaluated pregnancies ending in a live birth from 2016 to 2018. The VSD sites chart reviewed all cases with a diagnosis of chorio, and these cases were then clinically adjudicated by obstetricians on our team. We started this follow-up study in 2019, and then the pandemic hit, and our work shifted. We were able to finish these analyses and disseminate our study findings in spring 2023. In analyses accounting for vaccination and pregnancy to be a time-varying exposure, the adjusted hazard ratio for choreo following maternal Tdap was 0.96, with a 95% confidence interval of 0.9 to 1.03. However, we also found that the ICD-10 code for choreo had a low positive predictive value for actually detecting clinical chorioamnitis, but it had a positive predictive value of 81% for either clinical or histologic choreo. Findings were reassuring for other outcomes assessed. But similar to our flu vaccine work, the study did not differentiate between the two licensed Tdap vaccines. Our groups also evaluated inadvertent exposures to HPV vaccine in pregnancy. Data presented to the FDA preceding the licensure of the 9-valent HPV vaccine found in the vaccine trials, in comparison to trials of the quadrivalent HPV vaccine, there was a potential increased risk for spontaneous abortion when the 9-valent vaccine was administered within 30 days of the LMP, or pregnancy start. For our studies of inadvertent HPV vaccination and pregnancy, we compared women vaccinated either during pregnancy or peri-pregnancy to those vaccinated during a period 16 to 22 weeks prior to pregnancy, with all potential spontaneous abortion cases chart reviewed and clinically adjudicated. This slide shows our main findings from our paper published in 2021. We found no increased risk for 9-valent HPV vaccination in pregnancy or peri-pregnancy as compared to distal vaccination for any of the outcomes, including spontaneous abortion, preterm birth, SGA birth, or major structural birth defects. Of note, for all groups, the numbers of HPV vaccine exposed were low, and thus confidence intervals are wide. Both the pace and interest in our work on vaccine safety in pregnancy grew exponentially with the COVID-19 pandemic and authorization of the COVID-19 vaccines. It was around this time that the VSD and work led by our colleagues at Kaiser Northwest expanded our ability to identify and date our ongoing pregnancies in near real time through the dynamic pregnancy algorithm. In addition, many VSD sites expanded their capacity to integrate immunization information system data from their state registries into their standard VSD vaccine files. This slide shows the many studies our site has published on maternal COVID-19 vaccine safety, including the primary vaccine series and booster doses, with several additional studies in the works. In these last few slides, I'd like to provide some additional thoughts regarding post-approval surveillance of vaccine safety in pregnancy. First, regarding outcome selection, it's important to consider biologic plausibility when evaluating a potential maternal vaccine safety outcome. Is there a plausible explanation for how a vaccine administered during a given time period in pregnancy could cause a given outcome? When considering public health importance, is the outcome a potential concern for pregnant women or their providers, and perhaps fears related to this outcome pose a barrier to vaccination and pregnancy? Also, additional sharing of data from pre-licensure trials or other surveillance systems would be useful for guiding our outcome selection. I would also like to advocate that some important vaccine safety outcomes may not be well suited for automated surveillance studies and may require a different approach, with some examples shown on the slide. A second area I wanted to comment on relates to vaccine exposures in pregnancy. First, we learned after completing our Tdap studies that our findings could not be used in the vaccine label as we didn't evaluate outcomes by manufacturer. Starting with COVID-19 vaccines, we have included manufacturer in all of our analyses. Similar to my previous comment about outcome selection, additional sharing of data from animal or phase 1 studies could potentially inform our choice of exposure windows. And finally, now that we can incorporate state immunization information system data into our VSD data files, we can attest to its importance. At HealthPartners, we found 25% of our data in COVID-19 vaccines for our full VSD population were only found in our state immunization data. Third, moving forward, we hope that timelines for reporting results of our maternal vaccine surveillance work are realistic. We need time for vaccine data to be available and incorporated into our VSD files. We also need data used in the dynamic pregnancy algorithm that helps us identify and date pregnancies. We need for it to have time to mature. And finally, we need time for pregnancy outcomes to occur. With our work on COVID-19 vaccines, we were often asked to report on first trimester vaccination and risk for birth defects before these pregnancies were complete, before any potential birth defects could have been confirmed. And finally, it's critical when conducting post-approval maternal vaccine safety surveillance that analytic approaches to minimize bias should be used. Some common sources for bias are shown on the slide. It's also important to note that risks for outcome vary by gestational age of vaccination, and vaccine availability can vary by season. Furthermore, these optimal analytic approaches should be applied when data is incorporated into meta-analyses. And with that, I will end, and I want to thank the large team and CDC that supported this work.
Thank you to all the panelists. And so we're going to open up for discussion. I'll just see if anyone in the room has any questions to get us started with. There's one over to the back here. Yeah.
Hi. Yes. So I just wanted to maybe ask a follow-up on something that Dr. Chambers raised in her remarks, and something that I think many of your presentations might support, which is this idea of a national pregnancy registry. And I think sounds like a great idea in theory, has a lot of challenges in implementation. And so I'm curious if maybe you, Dr. Chambers, or others have thoughts around what that might look like, where it might live, who might staff that, and any other considerations. I know there's limitations, but just some initial thoughts on what that might look like.
Can you also share?
Well, that's
My name is Alex Hellman. I'm a senior program officer with the National Academies.
I think that's the million-dollar question. The idea, I think, is appealing for all of the reasons that we worry about single drug pregnancy registries being costly and having a hard time raising awareness in the population. But I think Jessica's presentation points to the complexity of doing this, and that's for one class of medications, let alone all. But honestly, I think we're kind of all doing something like that. It's just that it's not interconnected. So there isn't, other than somebody calling you up and saying, what are you guys seeing, and what you're doing compared to what we're doing, there isn't an infrastructure to link all of these, disparate things that are happening together. And I think it's doable, but and I don't know where it should sit. I'm agnostic about that. It should sit in heaven.
They have the resources.
Yeah, that's right. Yeah.
Right.
Yeah, I mean, there's certainly the funding aspects and the logistics of it, on top of, where does it even belong. But I think there's probably some steps, some, intermediary things that could be done now, just as far as, looking at common protocols and standardization of outcomes and the way that, outcomes are assessed and evaluated and coded, that would make at least, even pulling the data, even if they do sit in different places, if they're collected in a more standardized way, that could make them, more amenable to, meta-analyses, or, at least capitalizing on the numbers in a collective way through pooled analyses. So that's something that could be done on a much more, near future kind of timeline.
I have a question, but I was make one comment about that, is that, using common definitions seems like such an easy thing. We've been struggling for that for years across all of research, right? Not just in post-approval sort of spaces. And I think one of the things that you said, Christina, about hypertensive disorders, that seems pretty clear, right? We can see what hypertension is or not. But I will tell you, some of it is the reliance on clinical care, because it is hard for us to make the diagnosis, much less document it.
It's an evolutionary diagnosis, which makes it harder.
Exactly, right. So sometimes I wonder if we should be looking at treatment or medication use, right? If there's an antihypertensive used, they must have had a condition, but it might not have met the exact definition to then ICD, to meet the ICD coding. So some of those things that we need to kind of be thinking about as we go to some of these, other strategies, I think.
Something like that is done for hemorrhage in quantifying blood given, because you cannot estimate blood loss. I mean, the surgeon always says,
right, well,
or as close to.
Well, that's
you know?
because we're all great.
Right,
We're
right.
good,
Yeah.
sir.
So yeah, so using something I agree. I think there's has to be something more objective to measure that.
Yeah, exactly. Well, one question I was wondering, Dr. Albano and Dr. Chambers, you both of your discussions on your registries had really incredible follow-up rates, or I should say loss to follow-up being really low. Are there any successes or sort of pearls to give to folks as they're thinking about this, if they're endeavoring into these areas, research or data?
Go ahead, I'll add on.
You know, we've always said, and I believe this to be true, it's anecdotal, is that it's because of engaging the mom. So she's committed to taking part in this. That being said, not all people consent. But once consented, once they're in the study, they develop a relationship with the person who's calling them, the person who's collecting that information, and I think we're very successful then in retaining them in the study.
I would say, I mean, the antiretroviral pregnancy registry is in a little bit of a unique situation because it's anonymously reported by healthcare providers. So as long as they know the outcome, they are able to provide it. So there's less of a attrition due to loss of follow-up. With that said, I mean, in keeping the providers engaged and actually adding value back to them so that they are receiving information that's actionable to them and their patients and use in their practice, I think it contributes to that significantly.
Question.
Thank you. Wonderful discussion. This is Janet Hardy, most recently from Biohaven and Pfizer. So asking from the industry perspective, and since a lot of this translates into what goes into a PMR from the FDA, I love the idea of these disease-based large registries and efficient. How to make it happen. Jessica, the antiretroviral registry has run for an extraordinarily long time. That in itself is productive for information, but also daunting from an industry perspective. And I think, if I'm not mistaken, that that list you have provided of industry members, they have come and gone. It's not continuous participation throughout. Is that correct?
For some, they're, GSK being the originator, ViiV now owns all their antiretroviral products. So they have been part of it. Other innovator manufacturers, as they have developed their initial products and come to market, have been a part of it. Where we do see turnover is through the generics, because they do have a lot more volatility in the drugs that they have. Okay. So it does turn over to a small degree.
Okay, thanks. And in terms of baby steps along the way to the giant registry and have an idea, Tina, thank you. There is the possibility for some of the larger manufacturers to look at their product lists and consider whether there can be disease-based within or class-based within their own products and start looking at efficiencies and innovation from that perspective. But I also want to take this back to the FDA and ask somebody from the FDA to speak to what regulatory authority you have to be moving this ahead and asking for this type of requirement, since we're talking about PMRs and PMCs. Thanks.
I think that will be forthcoming in some of the, next presentations for sure.
If we sorted out the issue that we had in the first presentation, do you see if you have any questions that came in from virtual participants? I think we've got it sorted out. If not, I'll run something else up to you.
I don't know. You might be able to look at it and detect for me.
All right, I'm gonna grab it from you. Well,
Yeah.
the next person speaking,
Yeah.
I'm gonna grab it for you, and I'll get
Dr.
it just a
Claus,
second.
yeah. Megan.
Hi, this was really terrific, and it's so great to hear about vaccines, which you actually know when people get. Oh, this is Megan Close, a rheumatologist. It sounds like these different registries recruit patients really quite differently, and that all brings, pros, cons, biases, and I would love to hear some thoughts about how you all think sort of optimal ways to recruit patients and pregnancies into these registries?
I can start. So obviously with the pandemic, there was a lot of press information out there. People were very aware of what was going on. I mean, I think that's probably not the poster child for how to do this on a regular basis, but it did provide sort of a mechanism by which people were able to engage. And we still had a hard time getting like vSafe, the original vSafe platform up and going, getting notification out to vaccine vaccine centers, the drive-through vaccination places, the major universities, the other places, clinics where people were getting vaccinated. We provided communication tools, and I think there was a bit of a slow uptake for that. I'll say when I got my vaccines, I didn't see it. I looked for it, and I brought it with me the next time.
Yeah.
So I said, what about this? So we kind of had that availability. When we've been talking recently about how would you do something similar, not a registry, but how would we even engage people in using vSafe for RSV, I think one of the easiest things, at least on paper, would be to engage with prenatal care providers and just having that messaging going out and having QR codes that people could use while they're sitting in the waiting room for periods of time. Not that that's where they're going to get their vaccines necessarily, but it might be. I mean, there's that disconnect as well. Like the prenatal care places are not necessarily where these vaccines are going to be administered in those 10-minute appointments. So I think to me, there's an opportunity there, especially if it's a vaccine for pregnant people, to kind of partner with those groups and using the parent organizations to try to get that messaging out. But I don't think there's a super great answer. I think there are probably multiple pathways to try to get engagement.
I agree. I think we do direct to consumer because we tap into people who would otherwise never come to us. So we do social media, but we also recruit through healthcare providers, as you know, Megan, because rheumatologists, for example, are motivated to do this.
Yeah.
But we still target the mom for the reasons that I've mentioned before. I think. You mentioned the partnership. We're working on a partnership with the Maternal Fetal Medicine Network to help recruit participants into pregnancy registries for the reason you said, and also for the reason of trying to increase diversity, since that patient population represents a much more diverse population than who contacts us spontaneously.
Yeah, so I would add to that, in addition to kind of educational aspects for both physicians and patients, looking at distribution networks, and works particularly well in the case of vaccines. We ran a couple of vaccine pregnancy registries that actually tapped into military bases where they knew their vaccines were being distributed and were able to collect large populations of exposed women there.
Something on mine?
Yeah. Okay, go for it. So I would just ask you all, as individuals who have been involved in the design and the conduct of the studies, if there are ways that you would think about as key to how you might do it differently. You all talked about challenges and sort of ways, but if you could wave your magic wand, like what would you think about doing differently?
I'll say, and this is a that we create a really high bar for people to participate. So, it's great when they do, but we think and are working on some ways to do this, of testing different incentives that would appeal to people. Altruism is the major incentive why people participate, but that is probably not the only one, and we need to test those. And we also need to create a pathway to participation that doesn't require that you sit on 8 hours of telephone interviews and complete a whole bunch of paperwork. And so can we get what we call pregnancy registry light information that's informative, and maybe a subset gets the more in-depth information. So that's where I would see some opportunities.
I would say standardization and simplification. If you think about a healthcare provider who might be interested, even on the obstetric side, There are, if you look at the FDA website, I think well over 100, maybe 150 or even more pregnancy exposure registries available to report to. Well, certainly they're all going to have nuances with regard to what information they're collecting, how they are collecting it, what systems they are using, data elements, et cetera. So it's really a lot to ask for essentially voluntary work on already busy clinicians. So anything that can be done to streamline that would be very useful.
Right. I think the burden on patients, pregnant individuals, is significant, but the burden on providers, given all that we know about healthcare setting, physician burnout, and so forth, is a tough one to rely on as well. So,
Yeah,
Elyse?
and I'll just add, along with having the manufacturer data available so that we can always do the analyses by manufacturer, that I, would have loved to always have more time to validate outcomes before we study them. That often our approach is we conduct the studies, we think that we have good outcomes, and then when we have a signal, then we say, oh, was this a good outcome, and what was the positive predictive value, and then we do a validation study after, like we did for choreo. And so I also wish, sometimes we just said no and didn't get pushed to study outcomes where we felt like our data wouldn't work with this outcome, and just suggested other alternative approaches should be used.
Right. Yeah, and I think one other key thing there is, you're right. I mean, we had lots of conversations about the Tdap and choreo, and I think even looking at some of the data that we talk about as confounders, while it's important, to also look and think, well, why would we think that any vaccine is going to cause gestational diabetes, right? We know what the underlying causality of that is. Is it really a factor we should be even collecting or worrying about, right? If we have birth weight, you can see that there might be very significant LGA or something like that. So I do think we have to get smarter about what it is, because we can talk about all kinds of ways to incent providers, patients, whatnot, but at the end of the day, people want to be efficient and timely and still get their information. So I think as researchers and clinicians, we're going to have to figure out how to do that in better ways as well. So, all right.
One more quick question.
All right. Anyone out in the audience?
Just Lee Cohen, Massachusetts General Hospital and PI on the National Pregnancy Registry for Psychiatric Medications. Tina, I just wanted to sort of follow up with a comment that you said. I think we have an incredible opportunity to engage potential subjects digitally that we didn't have before. And frankly, during the pandemic, we found that our ability to procure medical records digitally, because we had to do it, we did it on the fly. And it was a terrific lesson that we were able to get quality data across a very large sample of women. So I agree with you. I think that we have to get creative in terms of really the issue of engagement as part of our recruitment efforts.
Yeah, in the way that people want to be engaged and to obtain information that we feel pretty confident you can get that way without having to sort of probe and
Exactly, and also collaboratively working with IRBs as we sort of move to using digital platforms to conduct the sort of science that's really going to inform clinical care.
All right. Thanks, everyone. Thank you all for your presentation.
Thank you.
I think we're
Thank you.